Hello, everybody. Let's cover the things. Welcome to this Acceleration Consortium first seminar of the month. We really want to bring basically the best people in the world that do AI, materials, drug discovery, automation. And of course, it is really an honor to introduce a very long-time friend of mine, Alex Zhavoronkov. I like to say how I met with Alex, because it's actually a cool story. Around the time we started doing AI for drug discovery in Boston, when I was at Harvard, I got this email from a guy, Alex Zhavoronkov, says, I want to meet with you. I said, I want to meet with you. And he could only meet me on the weekend. So I made an exception. He was so excited. So we got together in the weekend. And he told me, I'm also doing generative models for drug discovery. We saw your paper. I have a startup company. We were starting to work on this. And he convinced me that we should work together. And in a study carried out in silico, the first AI for drug discovery paper came out where generative model actually came up with a drug candidate in about 45 days. He probably will tell you about that story. Since then, his career has been meteoric. I mean, he has the lead in AI for drug discovery drugs as close to the market that they can get to date. I mean, we still have to pass a few hurdles, but he will tell us that story too. And what I like the story about Alex is that he is really coming at the field from a background of AI, and he was not shy of learning more biology than almost anybody in this room. Okay, so he really knows a lot about many aspects of biology, including aging. He's also, by the way, if you get to interact with him, a very fun and energetic person, dynamic, makes a lot of jokes. And so it is a really good pleasure to have him in the Acceleration Consortium. There's already a lot of collaborations going on with us, different aspects. We recently had a preprint online of a drug discovered with a quantum computer together with Alex, and a lot of cool things like that. And this is just the beginning. I hope that a lot of the people that interact with him during this visit and also beyond this visit make in silico medicine and Alex's team closer to the Acceleration Consortium in terms of all the cool things that will come out of our collaboration. So with that, Alex, I hope you have fun giving this talk. And what do I say about people online, Brandon? I guess they can enter their questions in the question box there, right? I think there must be like a question box or a chat or something. So people online, feel free to ask your questions there. We'll try to get to you as well. And with that, Alex, I think the floor is yours.
My friend, thank you very much. Please give me a hug. So everybody loves Alon, but very few people love Alon the way I do. Because I know how his story and how difficult it was. And now you guys have the best drug discovery person with generative AI and quantum at the University of Toronto. Proud to be Canadian. Actually, I'm not proud to be anything because nationalities do not matter and papers do not matter. Drugs matter, I think, quite a bit. But very proud to see what University of Toronto is doing at the intersection of drug discovery, machine learning, and robotics. It's truly revolutionary, and you guys are at the top forefront. So I'll be presenting some of the work that we're doing also with the University of Toronto, with Alon's group, and with some other groups through Alon's group. And I hope that it will help you maybe put some drug discovery questions into perspective, because research papers are great, proofs of concept are great, but when it comes to approved drugs, it's actually very difficult, much more challenging than a research paper. And now that I have gone through the time when you kind of go from theory all the way into human clinical trial several times, I realized that industrializing generative AI for drug discovery is completely different from publishing a fancy paper. And I'm going to talk about some case studies. I have a lot of slides. I have I think close to 100 slides. But I'll start with the most important slide, because this slide is important for everybody online, everybody here. Aging. This is maybe not a disease or a disease. We don't know how to classify this process, but every one of you has it, right? It's kind of like having a mechanism built into your body which makes you lose function after a certain age and lose everything and die. And even though it's a sad story, we need to realize that all the processes in biology and chemistry happen in time, at least at kind of at our level. If you go subquantum, maybe it's slightly different. And also everybody has age. So if you are using age as a feature that you are trying to predict with a generative system, the generative system naturally learns biology. So that was our premise when we got into drug discovery. And I'm going to talk a little bit about that. So if you want to understand life itself, you want to understand aging, because aging is the net totality of all pathological processes that transpire in time. And actually, some of those processes might not be directly pathological. And age is the most universal feature that is present in every organism. So if you open up the clinical data set, usually one of the features that is going to be the most abundant is going to be age. It might not even have gender or sex, whatever you refer it to, but it will likely have age. And age is the feature that kind of defines when you die. The higher the number, the lower the probability of survival. So higher chances of all-cause mortality. So we really want to teach AI systems to understand life in time. And also, aging research is a huge area that allows you to do drug discovery more efficiently. So aging research can be used as a platform of for drug discovery. And I'm going to be talking a little bit about how you can use aging as a platform for drug discovery. If you get interested in this topic, we run an annual conference. It's a 5-day event at the University of Copenhagen with my brilliant partner, Morten Scheibe-Knudsen, who took it from Basel to Copenhagen. Now it's the world's largest event, 160 speakers, 5 days, about 1,000 people on site, 7,500 online. And many big pharma companies there. So now we had Lilly, Novo, Regeneron, Biogen, Novartis, many companies present about how to approach drug discovery from the aging research perspective. Next year is going to be the same place, so but register early because it sells out very, very quickly. Usually it sells out several months in advance, even sponsorships sell out because the space is very limited. And this is a very good venue to discuss drug discovery with AI, because you'll have a lot of like-minded individuals. So a little bit about in silico. So we were born originally in Baltimore at the Emerging Technology Centers at Hopkins. And then we scaled globally. So now we still have a site in New York, but we're moving to Boston. We have a site in Montreal, Canada. So Daniel Polikovsky here is our CTO. He is based in Montreal, is supervising theoretical AI discovery and theoretical AI development out of Montreal. So that's where we have kind of AI strategy. We also have applied AI and some theoretical AI in Abu Dhabi. Very unusual place, but zero tax. And also it's truly amazing place to live. So high quality of life. And very cheap energy. So if you are training models, you are likely to get discounts on compute even from major cloud providers if you are doing things there. Asia is a global manufacturing hub. It also is valid for chemistry and biology. So if you want to do rapid chemistry experiments at scale with thousands of qualified professionals cooking chemistry, performing biological experiments, you probably want to be there because in my estimates, it saves you approximately a year of time. And that's a year of time to the patient. So it's very, very good place to be. And I'm very unhappy to see geopolitical tensions between different countries leading to deglobalization and longer times from zero to patient if you disconnect. So I hope that doesn't happen, at least in drug discovery. A little bit about in silico. So this is how we are organized. So think of us as having an in silico commander that runs a variety of AI tools. So we have our own model orchestra, so more than 700 different models, chemistry, biology, a lot of predictive modeling, a lot of models that are not AI, more traditional bioinformatics, but we don't care. Many of them have APIs, so you can talk to them. And a reasoning system can talk and launch those systems independently or in concerto or in sequence. We have an army of AI agent workers. So now we have, Daniel, how many agents?
600?
I thought it was more than 600. 600 was May.
Well, we'll have around 2,000 by the end of the year, so you can mark that.
Okay, so now we're somewhere between 600 and 2,000 agents that perform different tasks. So think of it as your lab mates who you can give tasks to, and those agents will go and perform certain tasks. We have a robotics lab. Now we're collaborating with the best group in the world in quantum. Hopefully, we'll be able to publish in a peer-reviewed journal. Right now, you can see some papers in arXiv. And we also published a few papers showing where quantum can be applied to drug discovery. What is important is we have software tools that are now used by 10 out of the top 20 pharmaceutical companies. So many pharma companies are actually using our tools at scale, and we don't get their data. We protect ourselves from their data, but we get proprietary user validation data. So they can either like or dislike the model performance, and you can get expert human feedback. So that's something that you really want when you have an AI tool out. You want feedback from the market to see if it works or not. And most importantly, we have our own biology, chemistry, and biologics expertise used for real drug discovery. We also are doing quite a bit of work on aging research. We kind of started as an aging research company. And now we're also working quite a bit on sustainability, on sustainable agriculture with Syngenta. If you go to our website and go to Innovation on Agriculture, you'll see how we collaborate. We now work also on CO2 capture, new materials, new energy chemistry, and lubricants. So we make considerable revenue now from CO2 capture projects with some of the companies that are doing research in this area. And I think that if you want to live longer, you probably want to live in a cleaner environment as well. And for us, we do need to focus on sustainability, not only on conservation. I think conservation is not going to help much, but on decarbonization and looking for new ways to clean up, also to facilitate for possible natural disasters. You want to have your climate control on the planet. So for that, we introduced in July I'm Gaia initiative, which kind of bridges generative AI for drug discovery and AI benchmarking for sustainability, along with the founding one of the founding fathers of this consortium. So one of the first scientists to join and provide some AI models for our consortium on GitHub. So please join. You can join individually and contribute models, data, or your AI expertise to the consortium. This one is currently nonprofit and not intended to be for profit. That's why we're not competing with, the Aramcos, Chevrons, etc. of the world. So we can try to bring them together because we're even in a completely different universe from them. We are, of course, a biotech. So most companies would recognize us as a biotech, and Fast Company recognized us as the most innovative biotech globally this year. And why? Because we have the software and platform only provided to the industry. So we cover biology, chemistry, medicine, and science. So in biology, we have a tool that allows you to do disease modeling and target discovery from scratch called PandaOmics. It's fourth iteration, so fourth year in the business, and a lot of people use it. So it's users all over the world create disease cards, omics comparisons using PandaOmics, and try to identify novel targets or prioritize targets for their criteria. Most of the criteria for target discovery are commercial tractability, novelty, and confidence, right? And of course, safety, durability, and many, many other criteria. But I think novelty, confidence, and commercial tractability are the three key questions that you want to answer for whether you want to go after a certain target or not. PandaOmics allows you to solve that problem. So for us, if you need targets, please let me know. We have like hundreds of targets for every disease. We have generative biologics that allows you to do peptide, antibody, nanobody. And those are commercial software, commercial strength software. It's very different from having an open source pipeline script, right? So it means that the software is validated. There are certain data security protocols in there and standards. We have generative chemistry, so Chemistry 42. That's probably the most famous tool that we have. So a lot of pharma companies have deployed it, and it allows you to generate small molecules with the desired properties. And then we have our chemistry module. It is binding the relative free energy calculation engine, so which is kind of similar to other physics-based models out there, but it's super fast, super cheap, and super accurate. So once you are done generating your molecules and you're down to, let's say, 20, you can use a much more computationally intensive tool to see if your hypothesis works. And whether your molecules satisfy your binding criteria. Have ADME profiling, golden cubes, and Nacho. I'm going to talk a little bit about that. On the biology side, our most interesting stuff that came out just recently over the past 2 years are precious models. Those are, I refer to them as life models. Those are multimodal, multi-omics, multi-species tissue that understand biology and time and allow you to do age prediction, target discovery, small molecule chemistry discovery, repurposing. So many different tasks using one model. So they are either transformer or diffusion based. And Precious 3, I'm going to talk about that at length. It's a multi-species model. So it's mouse, rat, monkey, and human. And it can do zero shot. You show a dog, it will predict the age of dog and also annotate its tissues, which is pretty cool stuff. On the medicine side, we have InClinical, which predicts the outcomes of clinical trials. We have Dora, which allows you to write research papers from scratch. And we have LLM assistants. We then validate those tools ourselves by turning them into valuable therapeutic products. So we look at kind of the shortest path from the problem definition to the clinic and try to identify the best target with the best novelty, confidence, commercial tractability criteria, generate our own chemistry, and go all the way to human clinical trials. If the target is too novel, so for example, if it's never been in clinical trials before, usually pharmaceutical companies would not want to buy it from you until you demonstrate a clinical POC. So this is paradoxical because very often pharma companies will come to you and biotechs, can you produce a novel target? You say, yes, of course. I'll say, well, what kind of, then you prove them, spend half a year proving them that you have this capability, show them the lab, show them the target, and then they say, oh, thank you, great, we have this capability, wonderful. Now let's collaborate. And then they give you a target to work on, and the target would be, I don't know, KRAS G12C, for example. So like, wow, so where is the novelty, right? So they're saying, oh, sorry, but yeah, we need some novelty, but for real programs, we want confidence. And that's kind of the trade-off that you will always be making. So if you want to have more novelty, you will have to invest more to validate, but then you can sell it for much more money. And we have a pipeline which works with targets on both levels. So we can go ultra high novelty. So when we picked TNIC, and I'm going to talk a little bit about that at length, when we picked TNIC target in 2019, there were approximately 100 papers mentioning the protein. So nothing in the clinic. Nobody had developed selective inhibitors, no selective inhibitors out there, and nobody tried to take it in the clinic. So when we showed this target to a few pharma companies, they're like, oh, sorry, it's not going to work. We're like, guys, sorry, but there are like 15 different AI models suggesting that it's likely to work in fibrosis and aging and other things. They say, well, sorry, but it's not, we haven't seen this. So great, so it's normal, let's go after it. And then we created our own chemical matter and took it all the way to phase 2, and now phase 2 is complete. So that's kind of my what I'm deeply proud of. We could have failed. We even started shooting a documentary movie about this program when we started it in 2019. So you can actually see in silico.com/docuthon, where we provide 500 hours of high quality footage at every step of the way, because most people don't even understand what drug discovery is, right? Some of the targets we go after, like USP1, for example, or TEAD, or CAT6, the level of novelty is slightly lower, the level of confidence is higher, commercial tractability is high, and we do those programs to validate our chemistry engine, for example, and also the ability to predict whether the target is going to be hot in the future and whether it's going to be efficacious. And then we sell those drugs to pharma. So USP1, when we started the program, it had only one front runner ahead of us in the clinic, no phase 2 data, and only limited safety data. And then there was one follower. And then the front runner was a company called KSQ Therapeutics. They licensed their drug in phase 1, I think, to a company called Roche. And ours was licensed to Exelixis right away, almost. It's a pretty big deal in our field in AI. So we sold this molecule for $80 million cash upfront, close to $1 billion in total value, and to a very expert cancer company, publicly traded, and they actually have perfect medicinal chemistry in-house. So it kind of shows you that sometimes if you pick the right spot for target discovery, you can make a lot of money to fund your entire kind of show for the rest of drug discovery and development. CAT6 sold at preclinical candidate stage to Menarini, $12 million cash upfront, over $500 million in total deal value. And others are kind of like that. So, FGFR2/3, I did this program for many reasons. One of the reasons is that you remember probably some people were criticizing our work on DDR1 kinase when we published, right? And those are the same people that had a competing company. And that's their front runner program, right? And I thought, well, once we realized that we have the capability to sell drugs and develop them at scale and fast, took us approximately less than a year to go from 0 to preclinical candidate. And the preclinical candidate used that company's molecule as a benchmark, so we outperformed in every category. So very quickly, just so that we can call it the revenge target, but we think that it's possible to sell it as well. Some of the targets, like for example, MAT2A, are targeting synthetic lethality, so MTAP depletion. And recently, IDEAYA Pharma demonstrated that they can achieve 39% objective response rate in a phase 2 clinical study with a MAT2A inhibitor, and suddenly it becomes hot, right? And before that, since some people saw toxicity in dogs, for example, dogs have different MTAP depletion profile. A lot of companies discontinued that program, and we are now there is IDEAYA and us in the clinic. So of course, this became very hot. So I just wanted to spend this time on this slide to explain to you the philosophies for target discovery. And there are as many philosophies as there are people. That's why there is no kind of cookbook for how to come up with a perfect target. Sometimes you actually pick the targets that are maybe not exactly hot, and then you need to take it to phase 2 yourself. There is no other way, and maybe in that phase 2 you're also going to succeed, but there would be some other competing philosophies for that disease that are outperforming your hypothesis, right? So very often you need to look at the market and do market analysis and understand whether you want to bet on the target or not. It's kind of like a molecular casino. And one of the reasons why in silico exists is that now I think that we have the capabilities that many countries do not have. So name like 10 countries that discovered a drug truly, from target discovery with their own chemistry, with their own phase 1, with their own phase 2. And there are very few. So in the Middle East, for example, when I'm presenting, they're saying, oh, great, but we can do it ourselves. Saying, well, show me one example, right? Sometimes their like highest aspiration is to run, an extended arm of a phase 2 study for a big pharma company. But previously, they haven't even aspired to go after de novo drug discovery. And de novo drug discovery, all you need right now is money and AI, because the rest you can outsource. So now you can basically, your world is an outsourcing machine, right? So imagine that acceleration consortium starts firing on all cylinders. People should come and outsource to you. right? So if a big pharma company wants to rapidly scale up and accelerate the drug discovery program, if all those promises that AC is putting out work out, big pharma should close their internal R&D, right? And new countries would be able to try and go and replicate, and you, for example, go back to the country you were born in, or migrate to a new country which just has a lot of cash. Take your expertise, use your acceleration consortium expertise, and start betting on drugs, right, with the cash that the country has. Why is it important? Because currently the FDA approves approximately 50 drugs annually. We did the analysis, about 35 are small molecules, maybe 7 of them are moderately innovative. So now think about this, right? Think about yourself as a patient 20 years from now. And your toolkit, when you get into the clinic, is going to be 10 drugs. None of them are curative. Hello? Do you want to live in this world? So we need to have more countries bet on this rather than invest, on weapons or something else, right? So there are so many people out there investing in technology of death, and so few investing in technology of life, right? And we wanted to demonstrate that it is possible for a small entity like ours without drug discovery expertise to demonstrate a case study where we can go from 0 all the way to efficacy ourselves and demonstrate something that most countries cannot do. So we started by developing a software platform, and many of those software tools that we are providing are published. So we try to make them as transparent as possible. And Pandomics has an application note and GCM. It has more than 60 target discovery philosophies, more than 25 different models. Some of them are AI, some of them can be called AI, but it's more traditional bioinformatics. User base is mostly biotechnology companies and pharma and academics. Pharma, at the end of the day, they can talk about novel targets, but very rarely they discover novel targets. And I wrote several papers explaining why they are not exactly contributing to novel target discoveries as much as academia. But it's the fact of life. So mostly it's biotech and academics that are coming up with really good novel target hypotheses, and they use Pandomics. It's priced for this segment. It's reasonably affordable. And for academic collaborators that work with us on a research paper, for example, we can make it free for a certain time. Generative chemistry platform allows you to generate novel molecules with the desired properties. So it has many generative models and many predictive models working in reinforcement learning fashion. Currently, it has 40 generative models. It will go down to just a few. Well, Daniel is showing that we're planning to have 6 models because now our multi-model capabilities are allowing to replace a lot of validated generative models that we have benchmarked. And those benchmarks are not just synthetic virtual benchmarks, it's all experimental benchmarks. And now we have also a chemistry platform, and 10 out of the top 20 pharma companies are using this tool. One of the advantages enabling us to move fast in software is our data warehouse, which creates a data lake of public and proprietary data. Proprietary data is our data that we generate in the lab, but the effect of this data is actually very marginal. So if you talk to big pharma companies, to cloud providers, to hyperscalers, many of them are saying, oh, your private proprietary data, that's your main competitive advantage. And then you need to ask them, okay, well, I have 50 different novel targets in-house. Here you go, with some level of validation. Go and buy those targets. Well, where is the competitive advantage? The competitive advantage is actually the ability to take those targets into clinical trials, right? And then demonstrate that they work. It's not your internal data. So we rely on public data. And my kind of philosophy at Insilico, we have several policies to ensure that we can work globally. We don't take government money, and we don't use private personal data. Even though I have my own kind of philosophy about personal data, you can get all of mine if you like, including my IPSCs. If you want to do any kind of cell experiments, all of that is available. I think it's an advantage to have your data out rather than a threat. But internally, we don't use personal data. We have multiple different data types in the data warehouse. Of course, we have a lot of chemistry, but we have omics, publications, QLs, patents, and grants. So we like to follow government money over time. So for example, grants are published several years before publications or patents, and they're always openly available. So if you follow the money, if you follow the grants, you can even represent the total amount of data you have in the form of money. So now we have more than $2 trillion of money in the data warehouse. So we can say that this is how much data I have, not in terms of number of parameters. Now we also have Chemistry 42 new update that we released on May 8th. Now next one is going to be in November. This new release also includes a tool called Alchemistry, which allows you to do DFT very, very efficiently, and ADME profiling, and also kinase selectivity. So we have many, many different new tools. We also have really good retrosynthetic modules, if you like to use that too. And now this tool is also available for academics in a SaaS format. Alchemistry is super fast, accessible, and accurate. So if you compare it with the current alternative approach, where I'm not going to say which one, but you probably know, it outperforms in most of the benchmarks. And alchemistry can be used in combination with generative chemistry on many, many applications. One of the applications is agrochemistry, so design of novel herbicides, pesticides, and material design. So it can also be used for carbon capture. Also coming soon, so now you will be able to purchase Chemistry 42 on the credit card. Is it done already?
Yeah.
Yeah, so we can already do it, yeah.
Chemistry and pandomics. You can just give us your credit card and buy it.
Yeah, not us. You do it, just try it. So, but with academics, we understand that it's much more difficult. Sometimes we provide grants and you can use it. And we like to collaborate with the top groups in the world, especially when the QLs that we collaborate with are also know what they're doing. Very few. So Alana is probably one of the very, very few. Now we also have generative biologics, which allows you to generate proteins, so peptides, nanobodies, antibodies with the desired properties. You can do either generation or screening, just like in Chemistry 42, and it's already available. So when I presented those slides last time, it was a few months ago. And you can see some of the demonstrated nanomolar activity for those AI-generated peptides from scratch. So we have done some experimental validation, and we'll have some papers coming up on that note. We also are working on foundational models. So we released one that is open right now called Nach0 or Nacho with NVIDIA. NVIDIA is the first author. And it's a natural and chemical language system. And Precious 3GPT, that's my kind of personal favorite. It's a life model, multi-model, multi-omics, multi-species, multi-tissue transformer that can predict your age, but also perform many, many other tasks. And one of the really, really cool tools that we just released is called Dora. Dora, I'm going to show you how it works. I hope you can see my screen. And I hope I'm sharing my entire screen. So if you can see it, you see the web interface. And let's write a new paper while I'm talking. Yeah. So it creates a research article for gene and disease, or research article where you just pre-prompt it and submit some of your current materials and write a paper. This is a multi-agent explorer. What's your favorite gene? Nothing like proprietary. Guys, come on.
Anyone say a gene?
Any protein you like?
Character is good.
Character
is good.
HER2 is right there already. Do you don't know any protein? Okay.
Oh, 22.
Which one?
F as in Frank, B as in boy, X, O, 22. Oh, the letter O.
Yeah. All right. What is your disease hypothesis? Where do you think is going to work? Cancer.
Overexpressed in cancer. Overexpressed in cancer.
Okay, any specific one or just any cancer?
Leukemia.
Okay, the blood cancer. So let's say leukemia. And here it should do it automatically. We're proving it. So FBXO22 inhibition, right? Let's say, works in leukemia. Then you can upload your bibliography if you like. So your favorite papers, mostly your own. Making a joke here. Then you go through a paper plan. You can do abstract, add stuff to it, right? Introductions, materials. You can put some research plan for yourself. But let's say we do it in fully automated mode. It may take about a minute to get to a research plan. So you can create an army of agents that will go to PubMed, that will go to Geo, TCGA, and other databases, start doing work for you automatically. And here you see that you can modify this research plan. So summary of known involvement, review of relevant publications. Let's add something else, maybe expression methylation data analysis. Let's say we want to add overexpression. So let's say review of expression in the brain. And whether and if we want a non-brain penetrating molecule. Okay, done, submit. And now it will go and launch this army of agents. You will see the bibliography is going to pop up with original references over time. And by the time I finish the presentation, we can go and review the paper. We'll be able to download it and send it to you. So you can see if the hypothesis is correct or not, but it should have good references. So in theory, it should improve your life as an academic. But now imagine that you also have a robotics lab. And in your research plan, you can pre-prompt it to go and perform some objective-based experiments if you're lacking some. And we actually have this capability as well. So now through Dora, we're trying to automate everything. So, and some of the data annotation tasks, some of the data generation tasks, some of the very rapid literature review tasks, summarization, analysis of already existing hypotheses can be done through agents. So think of it as an AI scientist, but in this case, it can actually help you automate many experiments. And $17 a month for friends of Alan could be much cheaper. So basically for the cost of compute, because we also use multiple large language models. That's
17 Mexican pesos.
What's the exchange rate?
20 to 1.
For you, it's fine. No problem. For you, anything. But now I want to talk a little bit about, so now, of course, in our field, very few companies manage to get into human clinical trials from generative AI, a lot of blah, blah, blah. And then they either die like fruit flies, or they fail to deliver, or they raise a lot of money and sit on this money, and then you just don't have the clinical programs. So I think that we have achieved a pretty substantial status in our industry, but I want to look back and see how it was. So in 2017 and 2016, there were kind of three groups that started doing Gen AI in drug discovery. So one was ours, another one was Alan's, and another one was Hongmin Cheng's at AstraZeneca. I cannot confirm or deny if I showed this history at one of those companies. These are the slides that are kind of genuinely prepared even for something else. That's why I'm always promoting Alan, because without you, this industry would not progress. Also, because there were so many naysayers in the beginning. You cannot even imagine, especially for folks like us who come from the computational side. into the realm of drug discovery. It's kind of like coming into the beehive and saying, hello, I'm your bumblebee now. That was truly amazing at that time. And I'm usually showing, this is probably like a little bit older slide, right? You probably have now, I don't know, 100,000 and 160 index. But I usually show this because this was the paper that went on archive in 2016. So not peer-reviewed, but October, I remember that. My hair almost fell out because we were in review starting July with similar idea. That was like the big reveal, right? So now whatever you do, put stuff on archive. We learned that. Hongming Chan from AstraZeneca, now he is in Guangzhou laboratory. He has his own big lab, his own company, and doing his own thing. But back then, he was part of Ola Enquist's group at AstraZeneca, and he published some of the first papers, molecular de novo design, through deep reinforcement learning. He also contributed to Moses, our consortium paper with Alan, and many others. So actually one of the true pioneers, and his team was publishing good papers, but without making like significant claims, but people by extension hated them as well. And Ola Enquist, he's still with AstraZeneca, to my knowledge, and he was on many of those papers, and they also started hiring pretty good postdocs to do frontier research in the application of generative adversarial networks and other generative tools for small molecule drug design. So very, very important. So of course, we also published a bunch. The most famous one is this one with Alan. I'm going to talk about that. So Moses, DrugGyan, and the cornucopia of meaningful leads. That one was actually submitted, I think, in July 2016 and published in December. But spent close to 6 months in review in the journal where it's actually easier to publish because people were failing to find reviewers for something like that, as you remember. And here I'm presenting this is 2017. And area for AI to I hope you can hear it.
And there are two ways to do strategies for AI-driven innovation in pharma to ensure that you get better molecules and much faster approvals. So one is looking for a needle in a haystack, and another one is creating a new needle. So our company developed a pretty comprehensive pipeline that utilizes both of those strategies. We start with a large numbers of molecules, large numbers of hypotheses, and then take it through a process that gives us very high quality promising leads that then later we validate and put into partnering and pharmaceutical drug development. But today I'm going to be talking about one technology in AI and deep learning called generative adversarial networks, which is essentially disrupting AI as well. It's a new technology.
So yeah, at that time we were already using GANs. That's I think March 2017. And it was easy to present at events like that, but medicinal chemistry conferences, they kind of laughed you out. So saying, oh, great, we can show a certain number of petals on the flower, but molecules are not like that, right? And you remember a lot of papers saying that AI is overhyped, blah, blah, blah. So if you look at some of the papers that we published, we started 2016 and culminated with Gentrol. Our first experimental validation was this one, entangled conditional autoencoder for de novo drug design. I think, Daniel, you were the first author, where we first synthesized the selective JAK3 inhibitors and tested. At that time, we thought that that target class was dead. We actually kind of commercially dead. We also had TYK2 inhibitors selective. We had JAK1 selective. So the entire kind of JAK family was covered by our chemistry. But we thought that those are not commercially tractable because they were not exactly novel targets. But then 2023, Nimbus sells TYK2 inhibitor to Takeda selective for $6 billion, $4 billion cash upfront. So it kind of shows you that sometimes going after not novel targets is pretty useful. But at that time, it was even more difficult to publish because people are saying, okay, JAK3s are out there. And then, of course, greatest bottleneck in generative is experimental validation. There was a recent paper published out of Tom Blundell's lab. We actually didn't follow that lab much before this paper got published, and we didn't see it before it got published. It came out in April 2024, and it shows you experimental validation for generative models, like real experimental validation. They have input, output, design task, target, hit rate, outcome, publication year. And for distribution learning, a little bit kind of older approach, because you really need reinforcement learning, there were multiple papers where we were kind of moderately present, but these are in silico papers where chemistry 42 or our earlier models were used. So we actually put a lot of experimental validation out there. And I'll talk a little bit about case studies. So 10 compounds designed using 100% automated generative AI being validated experimentally. That was one question that we answered with Alan. We asked this question with Alan, and we answered it with this paper. And this work was done in 2018 in the kind of summer, and then spent almost a year in review in Nature. First, went higher, then Nature rejected, got into Nature Biotech. Then a lot of rounds of review, but shrank to brief communication. But we still gave birth to this paper. Alan was the last author, I was the first author. And that's a very famous paper because we showed for the first time that we can use generative reinforcement learning. And at that time, kind of primitive approach to that, right? So we didn't have commercially tractable software. We used a bunch of models that we put out to design 6 molecules for DDR1 kinase. At that time, the level of novelty of that kinase was pretty low because there were already phase 2 studies on drugs for this kinase. The kinase is actually a much easier drug target class to drug. We got 6 molecules with Alan. So I always show this picture. This paper got very substantial attention when it was published, pretty substantial metric score. And a lot of people didn't like that attention. So nowadays, everybody is hyped out about AlphaFold. And of course, it's a big breakthrough. Show me like one preclinical candidate out of AlphaFold. I would love to see that, even though I deeply respect the tech. And in this paper, what we did, so we got a CRO who challenged us to a target that would be pulled out of the hat. They pulled out DDR1 kinase. We synthesized 6 molecules. 2 of them went into cell assays and metabolic microsomal stability assays. And 1 we synthesized just enough to do the PK in mice. All that in 46 days, including generation. So it's a pretty good kind of timestamp, right, to beat. So if you're acceleration consortium and your job to accelerate, well, try to accelerate this timestamp, right? But of course, to go after projects like this, you need to cut some corners, right? So novelty of the molecule was sacrificed because you need to have route planning out there. You need to be able to synthesize enough. Like we had chemists cooking chemistry over the weekend, of course, right? And much of that was done in China, where people take this kind of experiments very, very seriously, and they just did it overnight. But in 46 days, we managed to achieve that. And that paper was criticized immediately by some companies like Relay Therapeutics. We replied. So we got another Nature Technology paper that is now reasonably highly cited. So sometimes controversy pays off. And then at Unwise, last year in December, I attended an online seminar, and Abe was presenting. He was saying, oh, look, other companies like Exscientia versus Cyrus, Schrödinger versus Novartis, in silico versus QSQ. This is how they compare. So they do it fast because they do it in a similar way. At that time, he probably didn't realize that anytime you start a program like that, you file a lot of defensive patents around all kinds of chemical matter. Especially you want to, if your chemical matter works, you want to ensure that the companies that are trying to do fast follow very quickly, they are confused about what you are doing. So if you are not a great medicinal chemist, you will very quickly come to this, well, this is not our clinical molecule. So they shouldn't have compared it to us. It's like it's a different scaffold completely. But when you're presenting like this, like I felt very sad. That's why if you see my LinkedIn feed, and sometimes I was previously, I didn't like Adam Weiss, so to speak. Before that, we were friends with Abe. He was actually University of Toronto grad. And we were very friendly up until I saw the slide. Like, why are you doing this, right? Why are you showing that? But then, why are you showing that if your company, which started, 2 years before mine, failed to deliver a single phase 1 asset in 12 years, and your latest one is an IND enabling and it's TYK2. TYK2, there are already approved drugs, and right now, like you have to spend 4 years to get to the level where, and hundreds of millions of dollars to the level of Nimbus selling to Takeda. So, like if I were a venture capitalist, I wouldn't invest, or if I were a pharma company, I wouldn't give any upfront, right? I'm in the market. So why do this, right? Especially when you cannot guess my molecule. Well, now Abe is not at Atomwise anymore, recently, from what I hear. Yeah, so that company kind of is not doing extremely well. So I stopped doing LinkedIn rants. But recently, their kind of computational chemistry, who just departed, said that, well, Alex is bullying Atomwise like Musk or Trump. Bullying is never acceptable. And I'm just saying, just don't show my molecules compared to others, right? And yes, I think that your company is dying, sorry. And he is saying that, Silico has likely won the AADD race. Rather than being proud yet humble and being a shining positive charismatic leader, he keeps on beating and bullying opponents that are already on the ground. So it's very easy to pinpoint nowadays in the global community in this kind of Musk called the woke virus, but in our case, it's actually easy to label somebody as a bully online. If I'm just saying that, look, they failed to deliver a clinical asset in 12 years. Sorry, the emperor is naked. And you shouldn't criticize the other ones, right? So it's not bullying, and I'm not bullying specific characters. I'm just showing that, in the past, people are criticizing. And his argument actually for doing that, when I confronted him at this conference, I'm saying, why did you do that? We're friends, right? Are you going to be friends with me after this, right? No. Can I be your friend? No. Why would you do something like that, right? And he said that, well, because of that, those guys from Relay Therapeutics, right? So because of this paper, it means that anybody can bully us, I like this word now, at conferences, right? Actually, without us knowing. I wasn't supposed to be there. By the way, this is something that I just screenshotted today. So turns out this one went back. So this was like an IND enabling, and suddenly they reviewed and it's not an IND enabling, and Abe is no longer there. So yeah, I just wanted to tell the story, right? To explain that in the past, if you see some LinkedIn kind of posts from me about that company, not specifically about people, it's not bullying, it's just responding to that first argument. I didn't start it, I never start things, right? And that revenge target was done after people really made my life pretty miserable, right, in the beginning. So it was really, really sad to see that. So now, big question, what is happening here? Did they screw it up? I did, probably, right?
Stop sharing.
It stopped sharing. I will just share the entire thing, yeah. Perfect, now it should work, right? Yeah, it's sharing. We can check it if you like. Yeah. And now another question, can we generate compounds for targets without crystal structure? Another picture of Alan here with Mike Levitt, who is our advisor since 2014 when the company started. We published a paper in Chemical Science, which is now pretty highly cited, which shows that we took Panda omics used the PandaOmics data repository to identify a set of targets that didn't have crystal structure, but scored highly for hepatocellular carcinoma. Took a bunch of targets, one of them was CDK20, saw that the AlphaFold crystal structure that is predicted and does not, there is no experimental crystal structure, AlphaFold structure is reasonably good, gave it to chemistry 42, had to redo the AlphaFold structure a little bit with CompCam tools, gave it to chemistry 42. In the first round of synthesis, we got approximately 7.3 micromolar, and then we got to 180 nanomolar in the second round of synthesis, and went into in vitro validation and showed cellular activity. The beauty of this study is that if you look at the exact time of how long it took us to perform the experiments without, publication, etc., it took us approximately 50 days for 2 rounds. So you need to have benchmarks. So basically from 0 to in vitro with now novel molecules, and for protein without crystal, you can go in 50 days. And now this program's one of the academics that we didn't know in Hong Kong came to our lab and said, you know what, we were working on CDK20 and hepatocellular carcinoma for the past few months. Can we get your molecules? And we gave them the molecules. I think they're doing company now. We'll hope. Let's see if it works. So now can we generate compounds with the desired properties for a commercial clinical program? So here you need to be super confident that your drug discovery platform is going to work, because by the time you pull the trigger, you need to be prepared to spend $100 million, $150 million, and 5 to 10 years of your life working on this program to see if the validation, if the generation conditions are confirmed. So this is like risky stuff. This is where you bet your life. And I kind of decided to try. So one of the papers that just came out of our lab, small molecule TNIC inhibitor targets fibrosis in preclinical and clinical models, came out in March 2023. That's where we decided to actually push the trigger and try to go after end-to-end with a timer, but also with a lot of preclinical experiments. This one didn't get as high of an alternametric score as our first generative systems, because nowadays even a preclinical candidate is, another day in the office. Nobody is covering SpaceX launches nowadays because there are so many of them. But in this paper, it's super special to me because this is the first case where we demonstrated and published at every step of the way, originally blogs with data, but without having a peer-reviewed paper. So we showed how we discovered the target, how we prioritized it using hallmarks of aging assessment for the first time. I'm going to talk about that a little bit. Our own chemistry, 79 molecules synthesized. We looked at what indication it's likely to succeed in, performed many in vitro experiments, so 18 in vitro experiments and many in vivo experiments in multiple models. Lots of non-GLP tox experiments, and then we did GLP tox experiments. So by the way, GLP tox for one molecule would put you down at that time $3 million. Nowadays, it went down, probably will cost you about $2 million, right? So think about that. And even though it's 28 day, just the fact that it's GLP will cost you a lot, and will cost you like 7 to 9 months of your life. So during that time, you can actually do some indication expansion experiments if you want. If you really believe in a drug. And usually our preclinical candidate package includes 28-day non-GLP tox. So very rarely we fail in GLP tox. In this paper, we also disclosed a lung fibrosis preclinical candidate, kidney fibrosis preclinical candidate, and we also did an inhalable formulation and put mice on a nebulizer with the inhalable solution, showed that it works as well. In that paper, we used 8 healthy volunteers in Australia. And remember, that was COVID. It wasn't easy to enroll patients. But we demonstrated safety, microdosing safety. And then decided to do 2 phase 2s in New Zealand and in China, 78 healthy, 48 healthy in both regions. And demonstrated safety in 2 phase 1s. And all that in one paper. So since this paper was very difficult for reviewers to read, we even created a portal where you can converse with the paper in June 2023 when we were submitting. What is interesting about this target is that before, in 2022, we published a paper showing that we can rank multiple different targets by the level of confidence, druggability, and their implications in different biological processes like cellular senescence, extracellular matrix stiffness, genomic instability, that are driving the aging process, not only diseases. Of course, some of those hallmarks are also hallmarks of cancer, hallmarks of senescence, hallmarks of Alzheimer's. But we wanted to find a target that will score in many types of biology, a chronic biology, that if we address that chronic biology, we are likely to benefit in several diseases. And you can chat with this paper at papers.insilicogpt.com. By the way, super cool system if you're submitting to a bunch of reviewers, because sometimes they're like, oh, you didn't disclose the structure of your molecule. Like, well, go and ask the question, like, was the molecular structure disclosed? We'll say, yes, see figure 2. If you don't understand figure 2, go to supplementary figure 3. Here is a SMILES string. Ah, okay. But some amateur medicinal chemists, even after we published this paper, they couldn't really, on LinkedIn, they said, oh, but now I'm going to try to guess in silico structure. Great. So same as atom-wise story, right? So if you know drug discovery, and you are really going up to commercially tractable programs, you probably want to guess the right molecule, right? And in this case, it wasn't disclosed in the paper. But now what is interesting is that a week ago, we announced that we have completed the first phase 2 human clinical trial in 60 human patients, which is pretty cool. So it means that people with IPF is a deadly disease, 2 to 3 years life expectancy usually, very age-related. So people with IPF, average age is around 65. So that's why we wanted to select an age-related disease. And now we show dose-dependent efficacy. So the more drug you take, the stronger the response. And for the first time, we saw the improvement in forced vital capacity. So in IPF, you always see the decline. So on standards of care, you usually lose approximately 200 milliliters of lung every year in FVC. Here we started seeing improvements. And the more drug you get at higher doses, you get unexpected efficacy just after 12 weeks. So for us, from what we see right now, the drug clearly demonstrated efficacy. And of course, it demonstrated good safety and tolerability. So why this clinical readout is important is because there are many companies out there that are trying to do AI drug discovery. Many of them have repurposed already existing drugs, so either in-licensed the drug or repurposed the common reagent, or they took an already existing target with clinically approved drugs and tried to make a better molecule. In our case, we didn't cut corners. And actually, even those companies that failed to go after novelty, they failed to get into phase 2, and one just recently read out, and in phase 2, they didn't see efficacy. So showing efficacy is actually very difficult. So that increases the probability of success in other programs, right? So now pharma is looking at this more seriously, and it increases the confidence in the platform. So it means that the platform has created a valuable therapeutic that just completed phase 2A study with safety and efficacy. So sometimes it takes you 5 years to show that degeneration conditions can be confirmed. So that actually increases the confidence in the entire AI drug discovery. And since you are Acceleration Consortium, I'm going to skip a few slides because otherwise I'm going to be out of time soon. But recently I go to, can I cover this one? Because this one is like super fun. And this is a cool story. So I go to a conference called Longevity Summit in Dublin. I wasn't supposed to be there, but a friend of mine asked me to present. It's like a lot of people. And these guys, so Alex and Alex, from a company called Cat Longevity, approach me, and they have cat ears on their head. And I didn't know them before. And they said, Alex, can we take a picture with you? You're kind of our one of the favorite scientists. Like, okay, sure, let's take a picture. Who are you? Then saying, we're from cat longevity. We want to increase the longevity of cats. And we recently saw a paper out of Stephen Horvath's lab showing that methylation aging clocks can predict age in many, many mammalian species equally well with very high accuracy. And some of those species are cats. And within those methylation clocks for cats and for all species, TNIC score is in top 5. I'm like, what? And I had to rush to the airport, so I didn't have the time to attend their talk, but they sent me this, right?
Very interesting outcome. So one thing we were very glad to see, again, from Aubrey's conference is this very known target, TNIC, that was previously hardly druggable. And Alex Zhavoronkov in silico medicine just presented at the conference about how he managed to drug this target. It's a very interesting target because, for example, just 3 days ago, the Horvath lab published a maximal species lifespan association study from DNA methylation data, and TNIC is a very strong coefficient in that data set. So
yeah, these guys are actually pretty cool. They're from Oxford, both PhDs from Oxford, and it turns out that they're doing really cool research in cat longevity, and probably it's a good place to start because they don't live very long, but very cute animals, and people really like cats. So if we can extend their life, probably owners will follow. So watch this company. But they kind of put me in touch with those papers because there were 3 papers published last 2 years by Stephen Horvath's lab that you can predict age in multiple mammalian species, predict lifespan and maximum lifespan using methylation. But we and others overlooked the supplementary materials in those clocks, right? And there they took the CPG sites, connected them to targetable genes, well, to all genes. And if you do a little bit more research, you actually see that some of the targets like TNIC, KIF18A, KAT6B, those are in my pipeline. You've seen those, right? We found them using a completely different approach, right? Mostly expression-based approach, also using aging clocks and other target discovery philosophies. And they kind of scored highly in aging and age-related diseases. But now methylation clocks support this hypothesis, right? So when you see something like that, you get very excited. Some people kind of get excited when they see the starting point. But when you have a phase 2 complete program, you get even more excited. So that's why I am like super excited about this drug. This is a phase 2 a complete drug. I made a Tiffany green with a very specific shape. You can recognize it by just touching it. So it's very rare for somebody to have their own drug in hand, but also supported by aging research. Now I'm going to go very quickly. Do I have time or no? Okay. So can we repurpose already existing drugs using AI? And we partnered with a few scientists out of ALS, Answer ALS Consortium. So just like that, I presented at the conference at Hopkins in March 2022. Mary Chikowitz, the chair of neurology at Harvard, and Jeffrey Rothstein, her counterpart at Hopkins, invited me to present. And Lu Bai and Zhanke was there as well at this conference. Bai Lu is one of the most famous and reputable Chinese American neuroscientists doing really frontier research in neurodegenerative diseases. And since he also worked for GSK for many years, he actually has active therapeutic programs in phase 2 right now, several age-related diseases with 4B technologies. And Zhanke was the postdoc at Jeffrey Rothstein's lab, and then he went to Mayo Clinic, and now he has his own lab. He has a fly model of ALS, which resembles the ALS phenotype, and omics kind of resembles the ALS profile as well in neurons in fly eyes. So we took, right after this conference, applied for Answer ALS data. They have iPSCs differentiated into neurons from ALS patients. and also a lot of ALS patient data. We had a lot of primary data from ALS patients post-mortem in Pandaomics from public resources. We combined that into Pandaomics, identified a few novel targets and also drugs to repurpose for those drugs, and tested them in flies. Very rapidly published in Frontiers in Aging Neuroscience. We actually wanted to time it. So from the time we started collaborating in March to the publication in July, very little time passed, and we published a study where we showed 26 proposed targets. Out of those 26, you can see that 18 showed strong or moderate rescue of the ALS phenotype in fly model, and 2 showed mild rescue. And well, 2 made it worse, but the hit rate was there. So, and some of those targets we are not disclosing right now were picked by Lubai's lab and company, and they repurposed one of the drugs and put it into principal investigator-initiated study phase 2 in 64 patients. And the beauty of China is that many rare diseases are not rare, 1.5 billion people, right? So that's why I'm so against disconnecting from international collaborations in healthcare, because you have to access this patient population and do some studies quickly. In 1 year, they managed, under 1 year, they managed to enroll, and they will have a readout soon. So fingers crossed. You don't hear anything. It probably didn't work very well, but if you hear another announcement, it will mean that we have more than one target that passed phase 2. And this is a disease with no cure. But I think that this is the best approach for ALS, because then you can show that the drug is moderately safe, convince the PI, and do the phase 2 like clinical study with investigator-initiated trial. So if PI believes in the drug, they might try it on patients. And when the PI is famous and there is an abundance of patients with no cure, they would rather try that drug rather than, not doing anything. So I think that we need to replicate this again and again and again and again until we succeed. So some important benchmarks for you. So since you're an acceleration consortium, I would say this slide is probably the most important, so pay attention. If somebody were to show me this slide, I would be extremely excited. And if I'm a venture capitalist or a journalist or a pharma company, and if an AI company would come to me, I would actually ask them some questions, like, for example, how many preclinical candidates you have nominated out of your platform, and how many reached clinical stage? What's your average time to preclinical candidate across all PCCs? What's your longest time to PCC? What was the number of PCCs that you managed to nominate in a single year? So 2019, we went out and did our first big raise, $37 million for Series B. Sounds like a lot, but it's actually not. Like your phase 2 would probably be more expensive just for one drug. And you need to get there, and that's just for a CRO study, right? Excluding internal operations. And if you want to do research, it's going to be more. So you have to raise more. You have to feed the beast. So far, since 2019, we nominated 19 preclinical candidates. How many people here nominated a preclinical candidate before? 1. 2? How many nominated more than, let's say, 5? No? And how many took it into clinical trials? So usually at a big pharma conference, some people would raise their hands, but it would be a massive collaborative effort, and most of the time they didn't initiate the programs. So this is a pretty good result. 2 are in phase 2. 1 just completed phase 2A, safety and dose-dependent efficacy, so that's a big thing. And if you want to accelerate, try to accelerate this. So average time to PCC has to be 13 months. My longest one is 18 months, and that's the Nature Biotech paper, which explains how we did it. And you can talk to this paper at papers.insilicogpt.com. So that paper also presents our standard of quality for preclinical candidate nomination. So it shows you that we have 28-day GLP tox, and it has to be there. And in 2022 alone, we nominated 9 preclinical candidates. We actually think that our capacity is probably 12 currently, if we had unlimited amount of capital. So if we had, let's say, $200 million a year to spend just on research, we could probably get 12 PCCs. Just to put you in perspective, big pharma companies usually nominate 5 to 10 preclinical candidates in small molecules a year. So those figures are extremely important, and I wish I knew them before I started the company. So now I'm telling those figures to others, so you can actually try to set the expectations, right? So many people think that you can nominate a preclinical candidate faster, but try, right? Because even 28-day GLP tox, or non-GLP tox will still take you more than 28 days because you need to get the report, you need to analyze, you need to scale up. So it's scale up the chemistry for that. So it takes time, and that's kind of shows you the value of also international collaborations. So if we did not have CROs in geographies that now people are trying to present as their enemies, and maybe they are, but in healthcare they definitely are not. Without that capability, we probably would waste 1 year of time, just if we were to cook this chemistry ourselves. And even if we were to do it like in a fully automated way. So now another big question, and that question I don't see answered by many papers out there. And if you have like social sciences department, or if you have some business students at the University of Toronto who want to significantly contribute to the pharmaceutical research and development, I recommend going into pharma R&D productivity studies. So currently there are only few groups worldwide that I realized are actually thinking about that or doing the analysis. And then analysis right now is super damn simple. You go and ask ChatGPT, and then go and see primary sources, and you can probably publish in Nature Reviews Drug Discovery if you do really well, or Drug Discovery Today. And I would actually be reading those papers myself every time I hit the bathroom, just to read up on what people think about pharma R&D productivity. And there was one group that I highly recommend following. It's Sam Gellin Consortium, Alexander Schumacher and Oliver Gussman. So they publish a lot of papers now on AI pharma productivity and also pharma productivity. They now show that within big pharmaceutical company, the cost of one approval is $6.1 billion. And usually they do it over 12-year cycles. Crazy, right? So an internal drug discovery productivity within big pharma is significantly lower than when they in-license. So that is very, very important, right? But nobody is talking about target novelty, for example. How do you evaluate whether the target is novel? So if you show you the drug XYZ123 RBC, some number sign, and you've never seen before and has never been mentioned in literature, it probably will be called 100% novel, right? But usually people sequence some genes, right? So your gene will likely be mentioned in the literature several times or maybe many times, just lightly, maybe without any experiments. How do you call this target? Is it going to be novel? Probably still novel, right? Maybe close to 100%. But then once somebody develops chemical matter for it and touches it for drug discovery, then it starts losing novelty. And then when people start doing knockouts in mice, et cetera, novelty goes down. So you get more confidence. Right now, nobody is writing about that. So if somebody could create a framework for novelty assessment, that would be great. It would definitely help us a lot because we are thinking about this a lot. So we look at novelty using several parameters. Has it been in human clinical trials? If not, we consider it to be over 70% novel, right? That means that you can go and touch this, right? Within a year, you can develop a preclinical candidate. Within 2 years, you can be in the clinic. If nobody is there before you, you are first in class, potentially first in class. Is there a clinical POC? If it reached phase 2 complete, doesn't necessarily mean it's a dead target, right? If there are not that many players, you can still do best in class and still make a lot of money. There are many examples showing that the 8th attempt at a specific target results in a viable therapeutic with really, really good clinical parameters, for example, good safety, and you can actually still do really well. Other selective compounds, how many publications? So we usually look at that when assessing the target. And then for compound novelty, well, the question is it first in class, best in class and patentable, differentiated? What is the patent life? So there are many, many questions to ask for a compound as well. And again, there needs to be more work on the parameters. You put out even the criteria as a perspective, you will get 100 citations in a year or more. Within a couple of years, you can get maybe 1,000 citations. I will be citing your paper because currently nobody is working on that actively. So recently we started doing kind of target annotation cards, and I started putting them on LinkedIn. So you can find some of those cards and see if you agree with this or whether we should add more information on it. So we are assessing here, for example, KIF18A, really hot therapeutic targets. Currently, I cannot confirm or deny if pharma companies want to buy mine because it's potentially best in class, most likely best in class. So here we show the mechanism, chromosome, et cetera, Publications, 154. Is it a lot? Is it not? We don't know. Recently, in the last 4 years, 71. So half of the papers were published just last few years. Other small molecules or biologics out there? Well, not that many. And there are some companies that are taking it into the clinic, including ours. So currently, the leader is Velastra, phase 1, Genhouse, phase 1, in silico IND enabling. So we hopefully will get it into phase 1 this year. So 3 companies. Is it novel? I call it moderately novel, right? Because there is no clinical POC. And sometimes you actually want to go after targets like that because people, if this guys show phase 2, phase 1 complete, phase 2 complete, the target will become extremely valuable, right? Billions of dollars if you have best-in-class chemistry. Or if they fail, it's not going to be a huge loss for you, right? Because you'll spend a few $million getting there. But you can still, if you have best-in-class chemistry with very good safety profile, you can repurpose it to another disease for phase 2, right? That's another really cool thing. So thinking about target sciences and target strategy is a huge, important area. And one of the companies that is doing analytics in this is called Biopharma Trend. They recently did the analysis of novelty of different targets. And I highly recommend looking at this report, right? If you can do a better job, you should. And it will be highly cited, and you will get thousands of LinkedIn followers if you do this assessment. It's sometimes going to be more cited than coming up with a novel chemical entity and publishing in GMC. But you really don't know how good your AI is until you either get a regulatory approval. Well, in our case, we got the phase 2 now, right? But still, we're far. So until we get a drug approved and we show that it's best in class, we don't know if AI is good or not. Another one, sell several drugs to super expert developers that have your expertise and are preferably hunting for the same targets. So recently, as I've explained, sold licensed one drug to Exelixis USP1 and Menarini Cut6 for breast cancer for a substantial amount of money. And then this money that you get pay for more development, right? So my idea is to build a company that is actually not only sustainable, but it's also profitable. Imagine a profitable biotech. So we're trying to build a factory of drugs that is not only trying to accelerate the drug discovery process, but if I were at full capacity, I would double the amount of preclinical candidates that 2 big pharmas are producing right now every year, and with higher probability of getting to phase 2s. And some of these molecules, when you sell them, so this comes from public slides in December 2023 from Exelixis. They now call the USP1 XL309. By the way, Exelixis is a beautiful company. So if you want to see how a pharma company needs to be run, This is like the best. Their CEO knows every patient in every trial, every data in every experiment. He is a medicinal chemist by training, and I myself think that medicinal chemists are like superhumans. They know chemistry, biology, and medicine at the same time. So here he compared us to KSQ. Remember that atom-wise slide where they are showing that we have similar molecules? No way you can get to this kind of properties with similar molecules, right? And this is public, presented actually before Atomwise presented that fun slide comparing our molecules with KSQ. So they show that on many parameters like CYP inhibition, solubility, TOX, our molecule outperformed, and that justified an $80 million deal, of course, upfront cash, right? And there they also showed the structure, they showed the different combinations of drugs in different animal models, comparing us to KSQ's molecule, now belonging to Roche, and showing that in multiple preclinical experiments, ours outperforms. So AI-generated molecule can be significantly superior than a molecule that took multiple years of medicinal chemistry expertise, and of course, performing very well and is likely to reach the market. Let's hope if the target hypothesis works, and it can be vastly superior. So now we also thought about automating. Yeah, 5 minutes, I'm almost done, guys. Sorry, I'm a little bit over time. I can speak about this for many years, but I hope that it was valuable in terms of benchmarks. So if you learn the benchmarks, I wish somebody showed me those benchmarks before. And nowadays you think that, okay, well, automating the experiments will help you a lot. And we tried. So we developed a lab that goes from target discovery all the way to preclinical and translational research and can support multiple projects, predominantly working on target discovery, target validation. Nowadays, I can tell you that I have so many new targets that my grandchildren, if I ever reproduce, will be working on those targets. There is very little value in those new targets right now, right? So yeah, I can publish them. The most important application of the lab that we've designed is actually indication expansion. So that's where automated biology can help you a lot. So you can actually see additional signals from different cell lines, organoids, xenografts, tumor grafts, seeing where the drug may work. So this is my lab. Some of you have been there, I know. To see how we do things, and we would love to collaborate. It's in Suzhou, in the Bio Bay area. It's kind of like small Palo Alto of Suzhou. Highly recommend going there. Super green. It's lakes, beautiful area. I still prefer Shanghai, but Suzhou is also very beautiful, right next door, 1.5 hours drive. And we wanted this lab to look great. So we wanted people to feel like they work in the future. And we kind of got inspired by Tron and by Star Wars. And when I opened it, I had Star Wars music playing, and I kind of walked in a very specific gown. So this is how it looks like. So it opens up, recognizes your face, says ni hao. To your right, you've got dimmed windows. You can undim them, see the robots. And you can show the lab walkthrough with a laser pointer. So it goes and takes tissues, cells, organoids. Robot picks them up, grinds them, microplies them, checks them for mycoplasma or for fungus. Then I have an army of autonomous guided vehicles with micrometer precision. You position part of the sample into the incubator, part goes into the imaging station. 4 or 5 types of types of imaging, so CX7, Celigos, etc. Depending on how much sample you have, part of the sample goes into the omics lab, and you've got 3 types of omics: methylation, transcriptomics, whole genome. All that is processed by Panda Omics, and depending on your novelty criteria for targets, we'll pick some targets. Some targets will have tool compounds already, or what you call chemical probes. Takes them from the compound hotel, uncaps them, puts them on a liquid handler, picks the amount of compound you need, aliquots it, and then transplants that to the microplates with the original sample, incubates, and then repeats the process. So you get transcriptional, methylation, and phenotypic response, and you can very quickly assess how AI performed. AI gets either punished or rewarded, and at the same time, you get target lists. So you see where you got real hits. and where you get positive, false negatives from your hypothesis. And you get really good target lists for targets that don't have known chemical compounds. So right now we have a lot. So if you want to collaborate, pretty much any cancer you can imagine. Now I'm going to skip this slide, but just wanted to show you some really cool papers to have a look at, right? So where quantum can help you accelerate drug discovery theoretically. One paper that you should definitely see is our paper on arXiv, where Alan did a huge amounts of work. You have it right there. So talk to them. They are superheroes here in the Acceleration Consortium. And here for the first time, we've generated a bunch of molecules in a real quantum machine, ran them through the filters in Chemistry 42, synthesized and tested. And this is the first quantum generated molecule, which is super cool. And I'll stop here, but I just wanted to explain that aging is a very important area for research. And arsenic was just recently implicated in glucose metabolism and obesity. And another group at the University of Copenhagen showed that knockout mice with TNIC gene knocked out, they not only live, they also don't gain fat and retain muscle on high-fat, high-sugar diet. That's like Western diet. You can eat all you like and not gain fat and be muscly. That's my dream. So I'm not sure if it's going to work there or not, but I've got an inhibitor in phase 2. Imagine, right? And what I think is going to be the biggest next thing are life models. Life models trained on the entire longitudinal data from cradle to the grave, multiple species. So take a picture of this bioarchive paper if you like to collaborate. This is a multi-species, multi-omics, multi-tissue transformer that can perform multiple biological tasks, including age prediction, target discovery, compound repurposing. You can do differential transcriptomics and differential methylation profile generation. It can do a lot of kind of tasks with one model. So it's one big transformer. So happy to collaborate on that one, either on application side or developing this model forward, because I think that models that understand life itself are the future.